Okay, so good afternoon, everybody. So we are coming back as well to biology and to biology of development. So my team indeed is working on cardiac development, and today I'm going to show you, in fact, how we are very interested in the development of the cardiac valve, both in, I would say, for basic science and for physiopathology of the valve. So next slide, please. So, yeah. Oh, okay. So just to remind you the structure, the morphology of the adult heart. So we have 4 chambers, and because we have indeed these 4 chambers, we need valves. These valves are basically done or formed to direct the blood flow from one chamber to another chamber. So in the lab, we are very interested in the mitral valve, which is between the left ventricle and the left atrium that you see on the right side of the picture here, because in fact, this is one of the valves which is the most affected by disease, both genetic and acquired disease. So if we go back to basically the development and the development of the heart. So as you may know, the heart is a very interesting organ in terms of development because this is the first one to be fully developed and even functional in the embryo or the fetus. And it starts very early at the primitive streak at E7.5 when the cells from the mesoderm and the posterior region migrate to the anterior regions. And then they are going basically to join because this is a cylinder, so both sides are going to join and form what we call the cardiac crescent, because indeed it has a form of a crescent. So the red one on the picture is what we call the first heart field, and this is basically these cells, these early cells, early progenitor, which will give rise to what you can see on the right, to the left ventricle in red, and part of the atrium. And basically half a day later, there will be new cells which are going to migrate from another location from the mesoderm, still from the mesoderm. They're going to migrate dorsally in the embryo and form the second heart field in green on the picture. And these cells are much more proliferative, so they are going to give rise, basically, of all the other parts of the heart, so right ventricle of the outflow tract, part of the atria as well. So what about the valve? So the formation of the valve starts as well almost close to even 1/3 of the gestation, I would say, at E8, E8.5. And as you know, the heart is formed by the myocardium, by the muscle cells. And there is an inner layer, which is the endocardium, which is an endothelial layer, as well as there will be an external layer, which is the epicardial. So the muscle is between 2 endothelial layers. And the endocardium is very important for the valve because at E8.5, E9, this endocardium is going basically to delaminate, to separate from the myocardium, which you can see on the slide, the 1 to the 3rd square, basically. And then this is going to form what we call a cushion, so between the endocardium, which is delaminated now, and the myocardium. And this endocardium, at this very specific location, is going to undergo an epithelial or an endothelial to mesenchymal transition so that the cells can migrate into the cushion, invade the cushion, and start to form what is going to be later the leaflet of the valve. But one of the basic questions that nobody could answer for decades and decades is that as the endocardium is an endothelial layer, okay, so we are, well, next slide, but anyway, just to follow up, the endocardium is an endothelial layer, and the question is why there is an EMT process that takes place only in these discrete regions, which are what you can see on the left in the AV canal atrioventricular canal, which is the canal just between the ventricle and the atrium, where because at this stage there is only one ventricle and one atrium, and the other one is on the left part, the outflow tract, which is going to make later on the aorta. So there is, of course, an inner layer. The endocardium is everywhere in the heart, but just at these very discrete regions, EMT will occur. So what I was saying is that indeed, after EMT, after invasion of these cells to form the cushion, we have formation of the leaflet. Next slide. And these leaflets, as you can see here, this is a mitral valve here as well. So there are two, depending on the valve, there are 2 or 3 leaflets. But in the mitral valve, you can see here, for example, the septal leaflet, which is the leaflet which is so in at the side of the septum, which separates the 2 ventricles, and the parietal leaflet, or as also called mural leaflets, which is the leaflet which is aside to the ventricular wall. So what is very peculiar in the formation of this valve, this leaflet, is you see these cells are going to basically to arrange in the leaflets. So there will be still the endothelial layer, as you can see, endothelial cells, and then there will be a layer which has been defined more as an histology, depending on which extracellular matrix proteins we can find, which is called atrialis, and this is enriched in elastin, for example, and different type of collagen. And then you will have at the center the spongiosa, which is enriched in collagen and glycosaminoglycan. So there is indeed an arrangement of these cells, and we don't know so much, in fact, how these cells can arrange as a layer as well. So next slide. So how can we study during embryogenesis the formation of these valves, and how can we use indeed single cell sequencing to learn about the first process of this valve formation? So we use genetic lineage tracing to be able to track in these endocardial cells and later the valvular cells. So we are crossing a Tie2 crease. So Tie2 is a gene which is expressed in most of endothelial and cells, including the endocardium. So we cross Tie2 crease with a rosa tomato mouse, a reporter, so that we can very early label all the endothelial cells, including the endocardial cells. And then we can basically look at different stages, again, of valve formations. First stage, that I was just talking about 1 minute ago, E9.5, when there is only one ventricle and one atrium. So you see the atrioventricular canal, but in the picture with this square ADC, atrioventricular canal, so that will be able to dissect out. And you see, for example, the E16.5, all these tomato positive cells in the leaflet of the mitral valves. So next slide. So what we did is, in terms of protocols to have access to these cells, is indeed at E9.5 to dissect this atrioventricular canal, at E16.5 to dissect the mitral valve, in fact the annulus which supports this mitral valve. And we did the same at postnatal stage, basically at P0, because we wanted to get as well the neonatal valve. So we can dissect these regions very carefully in the embryos. Then we can dissociate in EEE all the cells. And when we can go to FACS, and we can sort out the tomato positive cells. And these are these tomato positive cells which are going to be processed in single cell RNA sequencing. And we use the 10x, as most of people now, 10x genomics facility. So next slide is showing basically a TCNE plots where you can see indeed the samples from the different stage of valve formation. you see all these black cells which are coming from the AV canal at E9.5. You see the pink cells which are coming from the mitral valve at E16.5. And you see the blue and gray cells which are coming from neonatal valve, mitral valve. So you see very clearly that the cells are very well discriminated. The AV canal cells very early on, so endocardial and valve progenitors. And then you see that there is a few overlap between E16.5 and the neonatal stage, where you see that some cells, indeed pink cells, are close to the blue cells. And what is very interesting here is the reproducibility, because, for example, the two samples of neonatal valve, blue and gray cells, you see fully overlapped. So they are very similar in terms of transcriptomic profile. So next, now we are going to go stage by stage to try to understand again the process of valve formation. So the first process, as you know now, is the EMT process, this endocardial to mesenchymal transition. We know now for a few years that the EMT is not a one-step process. The epithelial cells does not basically become a mesenchymal cells in one step, but in multi-step. And we know this from a lot of papers and a lot of research from the oncology field, as you know, because EMT is, of course, a process which is at the origin of metastasis in cancer. So people study this very extensively, and we know now that indeed there are different stages. So the epithelial cell is going to basically transform a little bit and to lose, in fact, connections and become what we call people call now early hybrid cells. And then the cells are going to change a little bit the shape, and this is what is going to be the hybrid cells. And at the late hybrid stage, then the cell will almost be mesenchymal cells, so they will be able basically to migrate and to invade different territories. So we wanted to look, next slide, yeah. We wanted to look if we can find the same genes as people found in oncology field, and if we could map indeed this EMT process of our endocardial cells, again, the first stage of valvulogenesis. So this is just a heat map showing that indeed you see from different clusters, we will see much better in the next slide, but you see that we can see indeed, for example, at the left, all the cluster of endocardial cells with typical endothelial genes like PCAM, like the V-cadherin that you see at the end. You see the endoglin ENG, which is as well an endocardial genes. And at the right, there is a very small cluster that you barely see, which are the valvular interstitial cells. So they are fully mesenchymal and they are already quite advanced in the differentiation. And between all these clusters, different clusters as you can see, are indeed the different stage or steps of EMT. Next slide. Okay, so to see much better how the cells indeed undergo EMT, so we use an inference. We use Scorpius, which is an inference method to be able to see indeed the progression in terms of transcriptomic profile of the cells. So this is what we call a pseudo time, and this shows very nicely the trajectory, in fact, of the endocardial to mesenchymal transition. Again, if you go from the bottom part of the slide, you see in red all the endocardial and cells, which are still endocardial, still epithelial. And then step by step, you will see in green and blue the early hybrid cells that are mapped by VCAM, what people again found in oncology. So they are highly VCAM positive. then you will see the orange or orange cells, which are low PKM, low endoglin, low CDH5. So meaning that they almost lost all the epithelial or endocardial genes, but they are now positive for fibronectin and one of the integrin beta 1.
I think we lost the signal from Michel.
Let's wait a little bit, maybe he will reconnect.
Yes. Oh, yes, he's here. Okay. Michel, could you please switch off your video? Maybe it will help. We do not manage to get him back. Oh.
Okay.
Okay, now great. You're even straight now.
I don't know what happened. And I can even share now. Okay, can you see my screen?
Yes. C'est bon.
Yes.
Okay. Can you see my screen or?
Yes. It's on the first slide.
Okay. So we were at the end of this trajectory. So okay. So in what I was saying is that indeed this pseudo-time trajectory analysis could indeed very nicely reveal the different stages or steps of EMT. And you see, for example, that many cells, which are still endocardial.
Michel, we lost the presentation. We don't see the slides anymore. We see your desktop.
Maybe it's better not to do the slideshow, to keep it as it is.
Okay.
No, we're still seeing your desktop. Okay, now it's fine.
It's fine? Okay. So what I was saying is that you still see these endocardial cells which are not differentiated and did not undergo EMT, as well that you see some cells which are assigned, in fact, the trajectory, like hemogenic cells, which are a part of the endocardiums, like any epithelial cells, which are endothelial cells, sorry, which are capable of hemogenesis. So then we go to the E16.5 mitral valve leaflet that we dissected. And this is something interesting, is that beside this AV canal that I was talking about, which is one of the main embryonic origin of the valve, there are some cells later on which can migrate into this valve, and they are coming from the epicardium. So this is what you can see here from again genetic lineage tracing that people did with a WT1, which is a typical epicardial marker, Cree mouse cross again with a reporter mouse, and we repeated in the lab this experiment. So this is definitively admitted now. And you see that indeed there are red cells here in the parietal, in the right parietal leaflets, but not in the septal leaflets. So indeed, the epicardium is capable to contribute to one leaflet, but not the other leaflets. And we use this in single cell analysis to be able to map indeed and to recognize a different type of cells. So these are the single cell analysis again from E16.5 mitral valve. And what you see in the TCNE plot here, that indeed we can map two different populations of valvular endothelial cells that we call VEC, and one cluster, which is the purple one here, very close to the green one, but in fact they share almost the same genes, except that these ones are indeed WT1 positive and the green one are WT1 negative. So meaning that already here you can see indeed the VEC, which are going to give rise to the septal or to the parietal leaflets. And we can see as well in this TCNA plot another population of cells, which are indeed the valvular interstitial cells. So the one which are really going to give rise to the different layers of the valve. And we could map these cells and track these cells because they express a lot of collagens and glycosaminoglycan genes. So this is again, this is a heat map of the E16.5 valve leaflets, which feature indeed the beginnings of valve layers, meaning that you see the different clusters here, this one, and so on, which indeed are this cluster show expression of genes which are going to be genes, again, which are more reminiscent of one of the layers, spongiosa, atrialis, and so on, enriched in one or specific extracellular matrix proteins. So, of course, the cells cannot be organized, but they indeed are already determined, if you want, in the layer in which they are going to be arranged later on when the valve are going to maturate. So now let's have a look at the single cell sequencing and the profile of the neonatal mitral valve. So we still recognize the two populations of VEC, the septal one and the parietal one, again because of WT1 expressions. And you see that they are very closely associated in this 3D TCNE plot. We see valvular interstitial cells, different population of VEC. Again, these are the population which now are more reminiscent and more differentiated toward a specific layer of the valve. And you see very separated for all these cells, CD45 positive cells, VEC, which are indeed, again, known to be present in the valve, even if we don't know yet, in fact, still, if they are coming all along the development of the valve, or if these are CD45 positive cells migrating later on into the valve. So, in fact, this single cell sequencing approach was very useful, and we combined this. I'm not going to comment at all, but we could combine this with a clonal analysis. So to do this clonal analysis, again, we use a reporter mice. So we cross the mice with a Cree, and the reporter mice is a multicolor, what we call a rainbow mice. It is multicolor. It expresses both red, the fluorescent proteins, a CFP, a blue fluorescent protein, and the YFP, the yellow proteins. All these, in fact, genes are flanked by different LOX genes with different sensibility to the Cree. So if we use a very low dose of tamoxifen to induce the Cree, we are going to basically get expression of one specific color. And if there are two different recombinations, we will have two colors or even three colors. But this is very rare because these are random recombinations. And you see, for example, at EAT 9.5 is heart, and this basically this AV canal, when you see cells in green, for example, and in red, which are coming from different recombinations. And then we could look at E16.5, and you see again all these clones that we have analyzed. So combined with single cell sequencing, and to sum up this first part, we can say that in this single cell sequencing allows us to track different biological process, specifically VEMT, but more than VEMT, which is very important to form the valve later on. And when we look at the cell which undergo EMT in the single cell, when we try to number these cells compared to the cell which did not undergo EMT, and when we did as well this clonal analysis approach, and we analyze by a statistical mean this clonal analysis, we could map that in fact we need about 40 progenitors in this AV canal endocardiums to build all the mitral valve. And these progenitors are capable to build both the septal and the parietal leaflets. So which is a very important information, and specifically to understand later on some disease when something is going wrong during the development of this valve. So indeed, going to the second and last part in terms of disease. So I was telling you that we are focusing on mitral valve because there is one of the disease which is very prominent, which is a mitral valve prolapse, which in fact is due to the fact that indeed this valve are going to secrete too much extracellular proteins, matrix proteins, make a lot of these proteins, so they are going to enlarge. And because they enlarge, they change of morphologies, and the leaflet, instead of closing properly, they cannot close anymore. So which means that there will be a leakage when the blood which is going to go through the valve cannot open and close very firmly, but it opens and doesn't close very well. And there will be, in fact, the blood flow can go back into the ventricle, which is a very bad prognostic for the functions of the whole heart. So in fact, one of the genes which has been found, there are few genes that we know now to be responsible for this mitral valve prolapse, and one of the genes is DAXUS, which is a polarity genes, and a mutation in DAXUS indeed is responsible for this mitral valve prolapse. So we decided to modelize this disease, and not using a mouse because it's very, very difficult. Even if in this paper that we did with collaborators, we could make a mouse model, but it's difficult to have all the different, to understand the mechanism, basic mechanism using the whole mouse. So we decided to use the iPS cells from patients who indeed suffer from mitral valve prolapse. And we set up a protocol of differentiation of these iPS cells into a valve, valvular cells, which is in fact a protocol that you see here. And you see again the process of valve formation during embryogenesis from the mesoderm, cardiac crescent, then the AV canal, and then the formation of the valve with the leaflets. And by adding different growth factors like Wnt, basically, and BMP, FGF8, VEGF to get more endothelial cells, and then using IBMP2 to trigger the EMT process, we were capable to get valvular cells. And these valvular cells are indeed all types of valvular cells. They are fibroblasts, periostin positive, they are chondrocytes, OX9 positive, or they are even scRNAseq positive, which are in fact the valve which makes this what we call chordae, which is the support of the valve, the mitral valve. So using this protocol, we differentiated the iPS cells from these patients affected by these DAXUS mutations and this mitral valve prolapse, and of course normal iPS from healthy people. And again, we did single cell sequencing of these cells when they are fully differentiated in valvular cells. And you see in blue the wild type, and in black the mutated cells. And you see very easily that these cells separate very clearly in this TCNE plot. And when you see on the right the different clusters which belong to these different two samples, wild type and Daxus mutant, you see that the wild types here are in fact not so much heterogeneous. There are only two main clusters, where you see that the mutated one are much more heterogeneous. So to make a long story very short, because a matter of time, I'm just going to show you the main feature and the main transcriptomic difference between these cells. And what I was telling you at the beginning is mitral valve prolapse is due to, in fact, a secretion, too much secretion of extracellular matrix proteins. So if we look, here we visualize different extracellular matrix genes, you see very clearly in these violin plots, or if you look first in the heat map here, again, you have in blue the wild type and at the left the mutated one. And you see different genes, all these genes are genes of extracellular proteins. You see a lot of collagens, luminol, periostin, decorin, for example. And you see that these genes are clearly enriched in the mutated populations compared to the wild type cell populations. And again, if you put many of the genes in the violin plot, you see that many of the genes are expressed in the mutated cells, they are much less expressed in the wild type cells. So to sum up all the story, I think you have been convinced that single cell sequencing in terms of developmental biology is very powerful to indeed get the different biological process. So here to build up the mitral valve, normal mitral valve, as well as diseased mitral valve, and which allow us, I didn't have time to point this out, but to identify new genes and new pathway which underline the development of this valve, specifically the patterning of the valve, again, the arrangement of the cells within these leaflets. And you see from the iPS cell model that the single cell could really be very powerful here to modelize and to better understand here valvular disease. So thank you for your attention, and if you have questions, I'd be happy, of course, to answer any question.
Thank you, Michel. It was a very nice and informative talk. Are there any questions in the chat?
I have a question. And Charles?
Okay.
Can you hear me?
Yes.
Head Charles.
So I'm curious, I might have missed it in your presentation. I'm sorry if I did, but in the, when you see the transition, the EMT process happening in the cells, so I guess the subset of cells is actually going to go through the transition out of the whole, whole developing organ. Do you know what triggers it, and is this, are these cells starting to migrate differently from the other cells? So in a sense, do you have more information about the kind of the spatiotemporal organization of the cells? Why do some cells trigger, and then what happens to them later? Maybe I missed it. You said it.
I'm not sure I understand very well what you mean. Why you are asking whether we could, from this transcriptomic profile, basically predict if the cells are going to undergo the EMT and thus to migrate into the cushion?
I'm trying to understand which cells, how do they decide that they are going to go through the EMT?
Well, this is a question I would like to know.
Okay.
In fact, what we found here, that indeed there is a specific transcriptomic profile of the cells which make them competent for EMT, but it's a little bit now like I would say the egg and the chicken is what indeed trigger this EMT, and why only these cells became competent for EMT. So one of the idea, but this is something we cannot see clearly from single-cell sequencing. We were, in fact, hoping to see that some cells could express specific receptors for growth factors, but this is not the case. We track them. What we have from single-cell sequencing, I didn't show you, but we focus on that. We have different genes of the metabolism, specifically a lipid metabolism, which are highly expressed in the cells which indeed were competent for EMT. So it means that in this endocardium, it looks like that there is an heterogeneity in term, I would say, of metabolism.
So if I think back at the pictures you showed very early in your presentation, you had specific regions in the embryo that go through the EMT. And so the cells that are in these regions, are they, I mean, I don't know if anybody knows the answer to this question, but are they there because they migrated there and therefore they're going to continue their fate, or did they?
No, this is, in fact, what I was saying is this endocardium, which is an endothelial layer, inner layer, is everywhere. It's basically everywhere in the heart. Okay, so in fact, this has been the question for many decades. Why the EMT, why this cushion form only in this very discrete region?
Okay.
Why this endocardial cells? There's a question, and this was indeed why we use single-cell sequencing to answer this question, to know if maybe from this transcriptomic profile, if they have indeed a very specific transcriptomic profile. So this is what I was telling just before. What we found so far seems to be related to metabolism.
Okay, thank you.
There is a question from Anne Schmidt.
Yeah, that's me. So I'm Anne Schmidt, working on the endothelial to hematopoietic transition. So it's something related to what you're doing.
Yeah.
So I was surprised to see that you can actually, so I don't know exactly of your conditions, how many animals you've been using and if they were well synchronized, but I've been surprised that you could really have these different populations of cells that are undergoing the EMT, basically different stages, because I would have expected, since it is a gradual phenomena, that it would be rather tricky to obtain the different stages. So can you explain a bit better how you manage to have these discrete stages?
Yeah, it's a good question. So in fact, we took many embryos, as you can imagine, to get enough cells, because we could basically calculate that in each AV canal of the embryo at E9.5, we have maximum 200 cells. Okay, this is what we did by fax and putting the cells in 96 wells. So we had to collect a lot of embryos, and we collect the embryos at E9.5, but then we select in the litter the embryo which have only between 18 and 22 somites. Okay, because you know that there is a lot of variability in development. So we try to select only these embryos. And E9.5, in fact, we knew already from the literature that this is indeed a stage when EMT has started already. So EMT probably start at E9, so half a day before. But as you know, it's very difficult to know if the embryo are at E9 or E9.5. We take the embryo only early in the morning. We decided to do like that. We take the embryo every time at between 8 and 9 in the morning. And the only way we had to decrease the variability is just to look at the somites and to select the embryo from the number of somites. So that's why we could probably get all the cells, and we selected for the E9.5 altogether at the end. We repeated this experiment twice. We had 120 embryos.
Okay. Okay.
And coming back to the first talk, from talking about to use fresh or a frozen sample, we have, as you can imagine, to use frozen sample because you could not get so many embryos the same days, as you can imagine.
I have another question, because the driver that you're using is expressing only in the heart, or more extended vascular system?
No, all the vascular. This is the type 2 crease, so it basically labels all the endothelial cells.
Okay, so then my question is, how can you make sure? So what did you do to really make sure that the cells that you are tracing performing the EMT do not come from another place? How can you make sure that they come from the heart?
Because I showed you the cell that we use, we first dissected in the AV canal, so in the heart, as well as we dissected the mitral valve in the embryo, only the mitral valve. So we cannot have any vessels.
Okay. Okay. Good. Okay. Thank you.
Okay. Are there any other questions? If not, then we will stop here for today, and I invite you to join through the same link tomorrow at 2 o'clock for the second part of the webinar. Thank you very much for the presenters and for the participants as well, and have a nice evening. See you tomorrow.